The biggest complaint most people have about FreeSurfer is that it takes so long to analyze a single subject. Like I said in the previous video, it can take 6 to 24 hours for a single subject analysis. If you have several subjects, like 30, 40, 50 plus, like you would in a normal data set, this can take days, weeks, just too long. However, even if you're using a single computer, you can cut down on that time considerably by using a command called parallel. I first found out about this on Pete Mulfese's website, and I've included a link to his blog post down below. To run Recon All in parallel, you will need to download the package Parallel. This requires a few prerequisites: the latest version of Xcode, which can be downloaded here, and the package manager Homebrew, which can be found here. Follow the instructions on the website to install Homebrew. If you already have Homebrew installed, run brew update from your command line to make sure it's up to date. To run Parallel, you will need to be in the bash shell. Type bash to load the shell. I like to set subjects.dir to my current working directory. The syntax for this in bash is export subjects.dir equals in backticks pwd. To run several jobs simultaneously in parallel, we will pipe in the subjects using the vertical bar. From the directory containing all of your anatomical Nifty files, type the following: ls*.nii, vertical bar, parallel --jobs 8, and then recon all -s, braces with a dot inside, -i, empty braces, and then the options all and qcache. Let's unpack what this does. Assume that we have 6 anatomical images in our directory, subject 1 anat.nii up through subject 6 anat.nii. The expression ls*.nii will grab every file ending with .nii. The vertical bar, or pipe, means to feed those file names into the following command. Parallel signalizes that the subsequent commands will be run in parallel on individual cores, and --jobs 8 indicates the number of cores to use. The recon all command is the same as in the previous tutorial, except it uses braces notation to make it compatible with the parallel command. Empty braces means to populate that option with the file list specified to the left of the vertical bar. Braces with a dot inside mean to strip the extension from the file name, in this case the .nii extension. This won't necessarily speed up the processing of a single subject, but it will run several subjects at the same time. For example, if a typical subject takes 10 hours to complete on your machine, then this command should be able to run 8 subjects simultaneously in 10 hours. My computer has 8 cores, which can run 8 separate jobs simultaneously. Check your hardware specifications to see the number of cores that you have. There are other options to save time, such as running your analyses on a supercomputer or even on the cloud. If you prefer to use your own machine though, Parallel can finish most datasets in a fraction of the time that it would take to process each subject individually. This frees up considerable amounts of time for playing Rock Band and eating Hot Pockets. Enjoy. Once Recon All has finished, it will populate the subdirectories for each subject with volumetric and surface data. Here's what the directory structure will look like for a single subject. The first 5 directories are the ones you will use the most. The last 3, I honestly don't know what they do. Here's what's inside those first 5 directories. The label directory contains labels, which you can think of as regions of interest. This includes the atlas annotations for that subject. The MRI directory contains volumes, such as the intensity normalized brain, the skull stripped brain, and the subcortical segmentations. The scripts directory contains records of everything that happened during Recon All. Nearly everything is in the file reconall.log. The stats directory contains structural measurements, such as thickness and volume, for each parcellation and segmentation. The surf directory contains surfaces, such as the peel and inflated surfaces. Today, we're going to look at the output of the MRI and surf directories using FreeView, Free Surfer's Viewing Program. Navigate to a subject's directory and type FreeView. Load the anatomical image by clicking on File, Load Volume, navigate to the MRI directory, and load the file t1.mgz. This is the intensity normalized anatomical image. This is the view panel. It contains the image you loaded. A 3D brain is in the center panel, and the orthogonal slices are in the side panels. You can click and drag the slices to update the side panels, or you can highlight a side panel and use the up and down arrows to page through slices and update the 3D image. You can select different montages to change the layout of the images. If you click on any of the slices in the menu bar, that slice is presented in the center window. If you click on the head, a 3D view is presented. This is the control panel, which contains a list of all the images you have loaded. As you add more images, you will see them appear here. Load another image, the aseg.mgz file. This is a segmentation of the gray matter, white matter, and subcortical structures. Notice that the aseg image completely covers the T1 image. Think of the images as layers. Whichever is on top will cover the ones below it. You can change the hierarchy of the layers by clicking the up and down buttons. Now the T1 image is the top layer and can be seen. You can hide an image by unchecking the checkbox, and make it reappear by checking it again. Also notice that highlighting an image will enable viewing options specific to that image. You can see these options in the toolbar located here. For example, if I wanted to change the color map of the aseg image, I can select Color Map and then Lookup Table. This loads a set of colors that correspond to the segmentations. You can lower the opacity with the opacity slider to see how the aseg image corresponds to the T1 image below it. Let's load some more volumes and surfaces. From the MRI folder, load white.mgz and brainmask.mgz. From the surf folder, load the white and peel surfaces, as well as the inflated brains. Highlight lh.peel and rh.peel, and change the edge color to red to better see the difference between tissue boundaries. It is important to view both the volumes and the surfaces simultaneously to make sure that the surfaces follow the expected boundaries on the anatomical images. You will be using this when we discuss failure modes, or how to repair data that was incorrectly parcellated or segmented during Recon All. Let's finish by looking at how to run FreeView from the command line. If you know what settings you want to use, you can save time by typing them from the terminal. The -V option means that the following images will be volumes. The F option means that the following images will be surfaces. Colons signalize which option to use for that particular image. For example, the color map for aseg.mgz will be the lookup table, and the opacity will be 0.2. See if you can figure out what the other options mean, and experiment with changing the options to make them best suit your needs.